everyone, welcome back to another class of Biomix-up. So in the last class we have already discussed about the glycolysis, about the aerobic respiration, what are the stages of this aerobic respiration. So today in this video we will discuss about the link reaction, how this link reaction is taking place, how it is linking the glycolysis and product of the glycolysis and the Krebs cycle. So at first what we will discuss here about the link reaction. Okay, so what happens when we discuss about the glycolysis, where it was taking place? It was taking place in the cytoplasm. The glycolysis which was present, its end product was one from glucose. Through the process of glycolysis, there was production of two molecules of pyruvate. Okay, so this pyruvate which is present, it will be transferred inside the mitochondria in the form of, and it will convert it into the acetyl coenzyme A. So here this pyruvate which is produced in the cytoplasm through the process of glycolysis, where it will be transferred into the mitochondria. So the mitochondria which is present, what it is having? It is having two membrane, like one outer membrane and one inner membrane. This is the mitochondria, and this is the mitochondrial matrix. Okay, so this pyruvate which is present, it is being transferred into the matrix. Two molecules of pyruvate is transferred and it is being converted into acetyl coenzyme A, and this acetyl coenzyme A is undergoing Krebs cycle. Okay, it is undergoing Krebs cycle. So what happens? From glucose there is release of two molecules of pyruvate through the process of glycolysis, and from that this pyruvate is being transferred into the mitochondrial matrix, where this 2 pyruvate is converted into two molecules of acetyl coenzyme A. And the process by which this two molecules of pyruvate is converted into two molecules of acetyl coenzyme A, it is only known as link reaction. Okay, and it is taking place inside the matrix, and it is responsible. So the end product of this link reaction, which is known as acetyl coenzyme A, it is responsible for the Krebs cycle to take place. Okay, so now what we will see how this is taking place, what are the components and what are the byproducts formed during these steps. So here two molecules of pyruvate is used. So this two molecules, this pyruvate which is formed, it is of 3 carbon and it is of two molecules, that is 2 into 3, that is 6 carbon is present over here. So this pyruvate, it is converted into two molecules of acetyl coenzyme A. This acetyl coenzyme A which is produced, it is a 2 carbon containing compound. So in total what we have 2 into 2 equals to 4 carbon. So what happens here? There is reduction of 2 carbon molecules. That means in this process, by converting the 2 pyruvate into 2 acetyl coenzyme A, there is release of carbon dioxide. Okay. So here in this case, there is release of 2 molecules of carbon dioxide, and there is requirement of coenzyme A. So 2 molecules of coenzyme A is being utilized over here, and 2 molecules of CO2 is being taken out. Okay, so in this process what happens? 2 NAD plus is converted into two molecules of NADH, and 2H plus is being removed out. So this whole process is taking place with the help of one of the enzyme which is known as pyruvate dehydrogenase complex. It is known as pyruvate dehydrogenase complex. Okay, or it is known as PDC. And this PDC which is present, it is having 3 components like E1 subunits like E1, E2 and E3, which is responsible for its conversion. Okay, so here what is taking place here? In this process, the dehydration is taking place as there is removal of water molecule, dehydrogenation is taking place, as well as here as there is removal of carbon dioxide, decarboxylation is also taking place. Okay, so in this case, when this E1, E2 and E3 are present, they are also having some of the specific names and they are having the cofactors. So let us see what are the, let us now see some of the components of the pyruvate dehydrogenase. The pyruvate dehydrogenase complex which is present, it was having three components like E1, E2 and E3. Let us see what are the name of this E1, E2 and E3. E1 is pyruvate dehydrogenase. Pyruvate dehydrogenase. Okay. E2 is dihydrolipoyl transacetylase. And the E3 is dihydrolipoyl dehydrogenase. So this all are having different type of function. So for its function, they also require cofactors. So the cofactors for pyruvate dehydrogenase is TPP. For dihydrolipoyl transacetylase is coenzyme A and lipoic acid. Okay, the next enzyme is dihydrolipoyl dehydrogenase. So the enzyme for cofactor for it is NAD plus and FAD. So they are having different type of polypeptide. They are having different number of polypeptides. So for this E1 and the cofactor TPP, it is having 24 polypeptides. For E2 it is having 24 polypeptides, but for E3 it is having only 12 polypeptides. So let us now see what are the function. If there are three different types of enzymes, so they must have different type of function. So the function of this E1 is for the decarboxylation. It is for decarboxylation and oxidation for pyruvate. Next for E2, it is the transfer of acetyl group to coenzyme A. Okay, so for this what happens, there is formation of acetyl coenzyme A. The next is the dihydrolipoyl dehydrogenase. So for this is what it is doing, what is its function? Its function is for reoxidation of dihydrolipoamide. Okay, so these are some of the functions. So here you can see this pyruvate which is used, and from this pyruvate there is formation of this acetyl coenzyme A, and for this what it requires, it requires a complex which is known as pyruvate dehydrogenase complex, which is having three components like E1, E2 and E3. Okay, they are the TPP, lipoamide, FAD, coenzyme A. Coenzyme A is also required. So these are some of the cofactors, and during the stage there is requirement of coenzyme A, there is removal of carbon dioxide, NADP plus is being converted into NADH, and there is removal of H plus. Okay, so during this time there are also regulation of this pyruvate dehydrogenase complex. So let us see how this regulation is taking place of this pyruvate dehydrogenase. So for its regulation, the regulation can be in two ways. Like the first is the covalent modification. First will be is the covalent modification. The second one will be allosteric modification. Okay, if we discuss about the modification of this covalent kind of modification, suppose that is a enzyme, and this enzyme which is present, it is having a active site. Okay, in its active site, if there is addition of phosphate group, there is some of the modification. When there is addition of the phosphate to the enzyme, there is modification of this enzyme. In case of allosteric modification, what happens? Here there is a site where there is active site, here there is active site, but in some other places if there is phosphorylation or some modification is taking, if there is phosphorylation in some other site after this active site, that means in the except this active site wherever the modification is taking place due to which there is modification of this enzyme. So for example, in case of covalent modification, if the phosphorylation is taking place, in the active site, okay, in the serine amino acid, during this time what happens, that is inactivation of the enzyme due to phosphorylation. In this case, the pyruvate dehydrogenase complex which is present in the E1 site, in the E1, what happens, specifically in the E1, when there is phosphorylation in the active site, phosphorylation in active site of the enzyme, there is inactivation of the enzyme. Okay, so what happens in the E1 complex when there is phosphorylation in the active site, specifically in the serine amino acid, there is inactivation of this enzyme. In this way it is covalently modified. Okay, so next is the E2. So in case of allosteric enzyme, what happens, when there is phosphorylation in some other site except the active site, then there is modification of the enzyme. This will be in the E2 case. In the E2 complex, this example can be easily explained. Like for example, we can see here, just a minute. For example, in the E2 complex, what happens, there is some of the regulations. Like in E2, the acetyl coenzyme A which is present, it will inactivate it or it will inhibit it, because as an acetyl coenzyme A is a byproduct. If this byproduct is already present, that means it will inhibit the E2. Why? Because this E2 was responsible for the formation of this acetyl coenzyme A. Next, the coenzyme A which is present, so it will work as an activator for this E2. Coenzyme A will work as activator for E2, but the acetyl coenzyme A which is present, it will work as an inhibitor for E2. So we must know about the ratios of this acetyl coenzyme A and coenzyme A. So if the acetyl coenzyme A and coenzymes A ratio is high, during this time what happens, there is inhibition of the E2. E2 will be inhibited. Why? Because here the amount of acetyl coenzyme is high as compared to coenzyme A, due to which the ratio is high. So if this acetyl coenzyme is high, that means it will inhibit the E2. Now the second case, if the acetyl coenzyme A is present and the coenzyme A is also present, during this time when there is high amount of coenzyme A and low amount of acetyl coenzyme A, during this time what happens, the ratio will be low. Okay, due to the low ratio, during this time it will activate the E2. E2 will be activated. Okay, this is how the ratio of the acetyl coenzyme A and coenzyme A is taking place. This was the case of the E2. Now we will see how the E3 is being regulated. So the E3 which is present, it is being regulated by the NADH and NAD+. The end product, what is the end product? The end product is the NADH and H+. When this is present, what it will do, it will inhibit the E3. But if the NAD+ is present, what it will do, it will activate the E3. So here NAD+ is working as a activator, and this NADH and H+ which is present, it is working as a inhibitor. Okay, if the ratio of the NADH, when we talk about the ratio, if the ratio of the NADH and NAD+ is high, during this time what happens, the E3 will be inhibited. Why? Because the NAD+ which is present, it is low in this case, and NADH is present, it is high in this case, due to which the ratio is high, so E3 will be inhibited. But if the NADH and NAD+ ratio is low, that means NADH is present in low amount, NAD+ is present in high amount, during this time E3 will be activated. Okay, E3 will be activated. So this is the work how they are regulating the pyruvate dehydrogenase complex, and this is all about the link reaction. So what happens in case of the link reaction? What are the things which we are getting as an energy product? We are getting here NADH. So when we talk about one molecule of pyruvate, when it is converted into acetyl coenzyme A, during this time there is removal of one NADH molecule. So here two pyruvate molecules are being used for the formation of two acetyl coenzyme A. So here how much NADH we will get? We will get 2 molecules of NADH as the energy product. So 2 into 2.5. So how much? It is the 5 ATP which we can get from this link reaction. Okay, I hope you have understood this. So here we will see how these enzymes are working. Like you can see here, there are 3 enzymes like E1, E2 and E3. E1 is pyruvate dehydrogenase, E2 is dihydrolipoyl transacetylase, E3 is dihydrolipoyl dehydrogenase. In this case, in the first case, in the E1, TPP is used as a cofactor. In this case, you can see here the pyruvate which is present, and from here there is removal of CO2, and this TPP is being converted into hydroxyl ethyl TPP. Okay, now this hydroxyl ethyl TPP, for its conversion into this TPP, during this time there is involvement of the second enzyme, or second step is taking place in this case. Okay, in this stage what happens? The transfer of 2 electron and the acetyl group from TPP to oxidized form of the lipoyl group of the core enzyme dihydrolipoyl transacetylase E2 to form acetyl thioester of the reduced lipoyl group. So here you can see here, during this stage when there is conversion of this hydroxyl ethyl TPP into TPP, during this time the oxidized lipoxylysine is being converted into acyl lipoxylysine. Okay, so now the third step. What is the third step? Third step is a transesterification, in which the SH group of coenzyme A replaces the SH group of E2 to yield acetyl coenzyme A and a fully reduced dithiol form in the lipoxyl group. Okay, so here you can see here the SH group which is present. What it is doing? It is the SH group of this. It is being replaced by the SH group of this coenzyme A, and it is being transferred into reduced lipoxylysine. Okay, so here it is being fully reduced. Okay, now the fourth step, that is the dihydrolipoyl dehydrogenase E3 promotes transfer of 2 hydrogen atoms from the reduced lipoxyl group of E2 to the FAD prosthetic group of the E2, restoring the oxidized form of the lipoxyl group E2. So here in this case you can see the SH group which is present. From here the hydrogen H group which is present, it is being given to the FAD, due to which it is being transferred into FADH2. And when it is being converted into FADH2 to FAD, during this time the NADH, this H which is present, this hydrogen which is present, it has been taken by the NAD plus to convert into NADH and H plus. Okay, I hope you have understood this. So let us again revise. So in this first case, this pyruvate, due to this pyruvate, there is removal of this carbon dioxide in presence of the TPP, and this TPP is converted into hydroxyl ethyl TPP. It is in the pyruvate dehydrogenase. So the next thing is how it will be converted into hydroxyl ethyl TPP into TPP. During this time what happens? The oxidized lipoxylysine is converted into acyl lipoxylysine. Okay, now in the third step, the SH group which is present, it is being transferred into acetyl coenzyme A to produce acetyl coenzyme A. In the next step what happens? The SH group from the reduced lipoxylysine is taken by the FAD molecule. The hydrogen from this is taken by the FAD molecule, and it is being converted into FADH2. When this FADH2 will be transferred into FAD, during this time the H molecules is taken by the NAD plus for the conversion of NADH and H plus. This is how the E1, E2 and E3 is working. So what is the name of the E1? It is a pyruvate dehydrogenase. E2 is dihydrolipoyl transacetylase. E3 is dihydrolipoyl dehydrogenase. Okay.